Welcome to This Explainer. Today, we're investigating the ultimate microscopic crime scene inside the human body. We're going to look at exactly what happens when things go terribly wrong at a cellular level. It's a fascinating and sometimes messy look into the microscopic battles happening right inside us and the evidence left behind when a cell meets an untimely end. Now, we all know cells die. It's a completely natural, necessary part of life. But there's a huge difference between a neat, scheduled retirement and a messy, catastrophic system failure. Today, we're focusing exclusively on those catastrophic failures. So, what actually happens when a cell doesn't just die, but dies badly, taking down the whole microscopic neighborhood with it? Let's find out.
Here's our roadmap for this investigation. First, we'll define accidental cell death. Then we'll compare necrosis to apoptosis. Third, we'll look at the mechanisms and morphology. Fourth, we'll dive into coagulative and liquefactive necrosis. Fifth, caseous and fat necrosis. And finally, number 6, fibrinoid and gangrenous necrosis.
Kicking things off with Section 1, defining accidental cell death, or what we can call the system failure.
The source material pins down necrosis using 3 absolutely crucial words: accidental, irreversible, and pathological. This isn't planned out. It's a biological accident, usually triggered by an injury or some kind of toxin. It's irreversible, meaning there is zero going back once this chain reaction starts, and it's always pathological, meaning it represents a state of disease. This is never, ever just a normal physiological process.
Moving right along to Section 2, necrosis versus apoptosis, or as I like to think of it, murder versus suicide.
Framing this as murder versus suicide really helps clarify what we're dealing with. Apoptosis is an active, genetically programmed suicide of usually just a single cell. It actually requires energy, or ATP, to pull it off. Necrosis? That's completely different. It's a passive mass casualty event. It doesn't even need energy to happen, and instead of taking out one isolated cell, it usually takes down a whole mass or group of cells all at once.
This really highlights the physical differences on a purely visual level. When a cell undergoes that programmed apoptosis, it neatly shrinks. It basically packs its bags and reduces in size. But with necrosis, the cell totally loses its ability to regulate itself. It just takes on water and balloons up, causing massive swelling and enlargement.
And, that swollen state leads directly into the next big difference: the cell membrane. In apoptosis, the membrane stays perfectly intact, just forming these little bubbles or blebs. That means zero inflammation is triggered. But necrosis is messy. Because the cell swells up so much, its membrane physically breaks. It becomes disrupted and leaky, spilling all its internal contents right out into the surrounding neighborhood. And that toxic spill directly causes inflammation.
Honestly, even down at the DNA level, the necrosis crime scene is a total wreck. If you run the DNA through an electrophoresis gel, which is a tool scientists use to sort DNA by size, necrosis just shows up as a random, shredded, diffuse smear. Pure chaos. Compare that to the highly organized stepladder pattern you see in apoptosis. Necrosis just lacks any of that biological organization.
All right, let's jump into Section 3, mechanisms and visual morphology.
The mechanism here basically acts like a factory exploding from the inside out. It's a precise 4-step cascade. Step 1, the denaturation of proteins, where the internal structural supports just collapse. Step 2, enzymatic digestion. This is where internal enzymes literally start eating the cell from the inside. Step 3 is that critical damage to the plasma membrane we were just talking about. And finally, Step 4, the disastrous leakage of cellular contents straight into the surrounding tissue. So, what does this actually look like? When pathologists examine this wreckage through a microscope, the visual evidence is pretty striking. First, the cells become densely eosinophilic, meaning they stain a deep, bright pink, which happens directly because they've lost their cytoplasmic RNA. Second, they take on this glassy, shiny look because all their glycogen stores are gone. And finally, they can literally look moth-eaten because the cell's own lysosomes have just chewed up its internal organelles. It's a completely ravaged cell.
Let's move to Section 4 and look at some specific suspects: coagulative and liquefactive necrosis.
It's important to realize not all necrosis looks the same. Depending on the tissue and the cause, the crime scene varies wildly. This table perfectly captures the stark contrast between our two most common types.
Coagulative necrosis is the most frequent overall. Its main mechanism is protein denaturation. Think of it like cooking an egg. It essentially cooks the cell, freezing it in place so the cell outlines are actually preserved. You see this in infarcts of all solid organs, like the heart, liver, and kidneys, as well as in burns and dry gangrene. But contrast that with liquefactive necrosis. Here, enzymatic digestion is the primary driver, totally liquefying the tissue so cell outlines are completely destroyed into mush. You see this exclusively in brain infarcts, plus fungal infections, abscesses, and wet gangrene. So solid organs coagulate, but the brain and bacterial infections liquefy.
Next up is Section 5, caseous and fat necrosis.
Caseous necrosis is a really bizarre middle ground. Our source defines it as an intermediate form of both coagulative and liquefactive necrosis. What makes it instantly recognizable to a pathologist is its very specific morphology. It has this crumbly, white, friable, cheese-like appearance. Actually, the word caseous literally translates to cheese-like.
Now, if you're wondering where you'd actually encounter this cheese-like tissue damage in the real world, the most common example by far is tuberculosis. It shows up specifically as caseating granulomas in TB patients. You can also spot this distinct pattern in certain fungal infections like histoplasmosis and blastomycosis.
Moving on to fat necrosis, well, the clue is entirely in the name. This specific type of cell death happens because of an enzyme called lipase. Lipase specifically targets and destroys fatty tissue, breaking down the fats and leaving behind these localized areas of destruction.
This usually goes down in one of two ways, traumatic or enzymatic. Traumatic fat necrosis can happen from physical trauma to fatty tissues, like in the breast. Interestingly, this resulting scar tissue can feel like a hard lump, meaning it often mimics a tumor clinically. Obviously, that's crucial for doctors to differentiate. Then you have enzymatic fat necrosis. This happens when the pancreas releases rogue lipases during acute pancreatitis, which ends up destroying the fatty tissues of the pancreas itself, the omentum, and the mesentery.
Finally, we arrive at Section 6, fibrinoid and gangrenous necrosis.
Fibrinoid necrosis is deeply tied to the immune system. Morphologically, it leaves this pink, fibrin-like deposit in the blood vessel walls. Pathologically, this is triggered by severe immune reactions, specifically type 2 and 3 hypersensitivity reactions. Basically, the immune system misfires and attacks the body's own tissues due to immune complex deposition. We see this striking pink damage in Aschoff nodules during rheumatic heart disease, in vascular conditions like polyarteritis nodosa, and in cases of malignant hypertension.
And lastly, we have gangrenous necrosis. Now, this isn't a completely separate pattern of cell death. It's more of a clinical term that represents massive multi-tissue failure, typically in a limb. As you can see, it's divided into two distinct types that link perfectly back to what we just covered. Wet gangrene is associated with bacterial infection, tying directly back to liquefactive necrosis. Meanwhile, dry gangrene typically comes from a loss of blood supply without an infection, which perfectly associates it with those preserved cellular outlines of coagulative necrosis.
So we've tracked the entire process of accidental pathological cell death today, from the swelling membranes down to the localized tissue destruction. And it really leaves us with a provocative thought. If a tiny local error, like a single denatured protein or a small enzyme leak, can explode into an irreversible mass casualty cellular crime scene, we absolutely have to marvel at the incredibly fragile microscopic balance keeping us intact and alive every single day. Thanks for joining me on this explainer, and keep questioning the science inside you.